Okay, let's move on to the next one. So this is a very famous famous tools for protein localization prediction. It's called MULoc. It also has already have two versions. The first one is published in 2017. It's actually the first deep learning method for protein localization prediction, and the citation already reached more than 800 so far. And the most recent version is MULoc 2, published in NAR web server issue, and we can just by look at this figure to introduce its architecture. So it's quite straightforward. You have the query protein sequences, and you will go through this large language model. Actually, you utilize the Proto-Trans language model given by the previous work, and they will get embedding from this large language model, and then it will follow some attention layer, and finally a multi-layer perception for the final prediction. So this final layer is for two tasks. One is the subcellular localization prediction, 10 localizations. And another task is to predict the sorting signals. So it tells you if this signal peptide, or if it is AGI anchor, or it is target peptides. So for attention layer, it also will produce some residual level interpretations. So it will tell you the potential important regions based on the attention weights. So we can see this MULoc web server tool through this link. So the submission is still straightforward. You just paste the protein sequence in this box, or you can upload some files of a file, including your protein sequences, and it allows the user some options. For example, you can choose the model if it's a high quality but slow, or you want some high throughput prediction, which is fast but less accurate. Also, you can choose the output format. You want all the output with figures. You can choose long output or short output, or short output, maybe just the summary without figures. So this actually takes quite some time for the prediction if you want to do the high quality. So I have already predicted some results in here. So you can see this lists all the prediction results for each of the proteins here. For this first one, it firstly predicted as endoplasmic reticulum protein, and the signals for that prediction, they think it is a signal peptide, and it found the potentially region for that signal peptide, which started from the N terminal of the protein sequence and lasted this long. So one common issue of the current attention-based interpretation method is we usually don't have a very clear boundary where this signal peptide should stop. So I think that's one future work for all the researchers in this field. And you can see it provided the probability for each of the class. The highest one is endoplastic reticulum. That's the reason it predicted that that class. And similarly for other things, this one is predicted as a plastid protein, chloroplastid, and the signal is a chloroplastid transit peptides. This kind of signal also reside at the N terminal. So you see this one lasts very long. So you can see this a long region have a very high attention score. And the next one is peroxisome protein with detected peroxisomal targeting signal. This kind of signal usually resides at the end of the protein sequence. One classical one is SKL. I think in our paper we also found this kind of signals. So yeah, so this is the information provided by the MULoc tool. Very straightforward, very simple. The prediction, the signal, and the region. I think the some unique things for this method or tool is that one is that they are the first, they are the pioneers in utilizing deep learning in protein localization prediction. And this group, like I said, they also have their own system. So this MULoc series is for general protein localization, and they also have other branches. For example, the target P for target peptide and the signal P for signal peptides. So they are a group of people that are dedicated in this field for many years. And by reading their paper, you will see the way they do things is very clear. So that's the reason their database, that their data collected is usually used as a benchmark by many other groups. So for the prod-trans large language model I mentioned previously, when they do this localization task, they also benchmark on their the MULoc data. So you can see one thing we can learn from them is that you don't have to do very complex or fancy research just by doing things very in rigorous standard and do things very clear. You can also get your work settled many times. Okay. We can. So I think I have introduced many tools from other group. The last one is actually a tool from our group. We call it MULocDeep. Oh, sorry. So our group has also a relatively long history in protein localization prediction research. We have previously published a tool called MULoc that is a tool specific for plant mitochondrial protein prediction. So we predict if a plant protein, if it is a mitochondrium or not. And recently we published this MULocDeep. That is a deep learning version of protein localization tool for 10 classes. And in this year, we also published a web server for this method, published in AR, a web server issue. And this is a workflow of our method. And we also just require protein sequences for our tool. And the protein sequence will go through our method, and the results we provide to users through our web server are rich. For a single protein sequence, we predict what's the localization in these 10 subcellular compartments. And we also have these residue level predictions based on attention score, attention weights, and you can see which region is the potential localization signal. And also, besides the 10 subcellular compartments, we uniquely provide this 44 suborganellar compartments prediction. So this is the most comprehensive suborganeller level prediction so far. So and this result is consistent with the subcellular level. So if you predicted this protein is nuclear, you will see inside the nuclear or what's the subnuclear localization for this protein is predicted. And so we also uniquely provide some prediction results for protein groups. For example, we calculated the average attention scores for protein groups and draw this position weight matrix. So we'll know, for example, you give, for example, our tools predict a bunch of proteins as mitochondrion, and we will calculate the average attention scores for this mitochondrial groups. And the benefit is that for single proteins, usually this attention weights will affected by some noise. But if you have a bunch of mitochondrial proteins, it will reduce the bias or noise. So it will have a much better signal visualization for the signal peptides or target peptides. So here is an example. You can see we can tell that this region has a relatively higher attention weight than the rest of the sequence, and you will also see the proportion for each residue. What's the dominating amino acid on this residue? And another function for protein groups is that we calculated the attention weight ratio. This is the average attention weights divided by the attention weights from a randomly shuffled protein sequence. So if you got a ratio nearly to 1, that means your protein groups doesn't have any signals related to protein localization. But if a ratio is like this, so have a region much higher than 1, that means this region is potentially have a signal related to protein localization. So we will do a demo using our tools. Okay, this is the main page of our localization prediction tool. Also, we have this submission panel here, which is very straightforward. You can either paste your sequence or you can upload by a file. And we allow user to choose the kingdom if known. Otherwise, you can just select unknown. And you can give your job a nickname. This is just for easier search later. And you can give your email address if your job takes quite long times, so you don't have to wait here. We will notice you when the job is done. And we have this news panel here. It tells you some updates of our tools or some related activities. For example, we gave a workshop last year, so you will get noticed by looking at our news. And you can contact us if you met some issues using our tools. So this also have this map here. It tells you how many users, how many proteins has already processed by our tool. And clicking it, you will get some details, the information for the user usage statistics. In other tabs here, if you have previously submitted a job, you can trace back your job by typing the job ID you just gave in the submission page, or you can. Oh, sorry. You should use this nickname here, but we automatically assign a job ID. It's a series of numbers. You can search your job also by that, and also you can search by your job by the actually actual protein sequence in your job. And we can load an example sequence here. It contains 5 proteins, and then we can submit the job. While we introduce our tool, we can wait for it done. So here is an example. In this example, we have a lot of have maybe less than 100 proteins. The reasons is that we want to have more proteins so we can test this realization for protein groups. And you can see some proteins are grouped as nucleus, cytoplasm, secreted mitochondrion. And from our knowledge, we know that usually we don't have a signal peptide for cytoplasm proteins, but we do have such signals for secreted or mitochondrial proteins. So we can check the realization for the protein groups, attention score, or the ratio. So you can see for cytoplasm proteins group, the attention ratio is near 1. That means for both N-terminal and this is for terminal, the C-terminal, sorry. That means we don't have we don't detect any sorting signals here. But for secreted or mitochondrial proteins, we see this ratio is has a region that has a very high ratio in N-terminal, but for C-terminal, we don't have such high ratio. It's near 1. That means this signal only exists at N-terminal. Also for mitochondrial proteins, we also found some high attention ratios in C terminals. So this is for protein groups. For each individual proteins, we also provide several prediction results. One is the subcellular prediction and suborganeller prediction. So if you predict as a nuclear protein, you will see inside this nuclear what details localization are predicted. So in this case, it is a chromosome protein. And we can also see the residual level prediction. So for nuclear proteins, usually this signal can be anywhere in this protein sequence. So maybe we can find another protein that should be at the N-terminal. For example, this is secreted protein. You can see the signal is much higher in N-terminal. So it consists with our knowledge, and you will see maybe this one is a margin because this sharp reduce of attention weights. Okay, so other things we have in our web server is that you can check your the status of your previously submitted job. You can see all the jobs are done, and you can also do some operation on those jobs. You can either delete them or you can go to the results. So in this one, we only predicted the 3 proteins and all of them are mitochondrion. And we can only see the visualization for protein groups if that group has more than 10 proteins. So in this case, we won't see that kind of realization because it's only 3, but we can still get the prediction result for individual proteins. And in this last tab, we can see some other tools. For example, our previous MULoc, you can still access that if a tool there. Okay, maybe the server has some issue recently. And also for other resources, you can download our standalone Docker. You can use it free academically. and some you can also we can also cooperate with collaborate with some other companies for commercial license. We also have another version, it's a corner version, so you can also test it our tool locally in your computer. Also, okay, I think that's the demo of our MULocDeep. So finally, I want to use these slides as a summary. So you can see there are a lot of tools for protein localization predictions. So the main question is which tools you should use. So it really depends on your own research target. For example, you can if you are so some different tools have different coverage. So you should choose the tools that covers your interested compartment. So in our review paper, we have this table tells you what tools are covered what compartment, and we have the coverage code here. And also you can, if you have other needs, for example, you want to know the target peptide region or the signal peptide region, you should consider tools that give you such information. For example, the test detected the potential target peptide region and tells you the exact cleavage site of that target peptides. So we also notice annotated such information here. And also, you should choose tools that are based on your studying species. So include the eukaryotic protein, eukaryotic species or prokaryotic species. Also, some people are studying a virus or bacteria, so you can also choose tools accordingly. And the last thing we list here is their web server URL. And if you want to use these tools in your local machine, you should consider tools that provide a standalone version. So that's some information I think we think useful for users to choose their protein localization prediction tool. Okay, that's the end of this demo lecture. If you have any questions, I'm happy to answer it. Thank you.